Hello, I am Sylvia Azania, and in this video, we are going to talk about embryonic culture in the IVF laboratory. We will discuss when it takes place, the stages of embryonic development, how this culture should be, and we will also answer the most frequently asked questions related to this topic. The laboratory culture of in vitro fertilization begins after obtaining the eggs through follicular puncture. The day of the puncture is known as day 0 of embryonic development, as this is when fertilization, the union of the egg and sperm, will occur. Initially, the eggs will be cultured without removing the layer of granulosa cells surrounding them, a process known as decumulation. However, depending on the IVF technique that will be applied to fertilize the eggs, the next step will be, if it is conventional, IVF. The eggs are combined with the sperm without decumulating. If it is ICSI, the eggs are decumulated and then the intracytoplasmic sperm injection will be performed. The cultivation of embryos obtained after fertilization will continue until the day when the embryos are transferred to the maternal uterus, which is usually on day 3 or in the blastocyst stage, day 5 to 6, are vitrified, usually on day 5 to 6, are discarded for not continuing their development. It is worth noting that those remaining embryos that are vitrified can also have different destinations: future personal use, donation for research, transfer to other couples for reproductive purposes, or cessation of their preservation when the woman's reproductive stage has ended. If you would like more information, you can visit the article I have linked in the tags and in the description. And before we continue with the content of this video, I'm going to ask you please, if you're enjoying it, to give it a like and subscribe to the channel if you haven't done so already, as it helps us continue and ensures that all the information we publish reaches more people. Next, we will briefly review the stages that embryologists evaluate to select the embryo that is most likely to implant and therefore to result in a pregnancy. Day 1. This is the day after the follicular puncture, and it is when it is assessed 16 to 18 hours after fertilization whether it has occurred correctly. At this stage, the embryos are called zygotes. Day 2. The embryos have already undergone the first divisions and have 2 to 4 cells, also known as blastomeres. Day 3. At this point, the embryos have about 6 to 8 blastomeres. The embryo is beginning to express its own genes, so its needs are gradually changing. Day 4. The embryo is in the morula stage and is compacting. Days 5 and 6. During these days, the embryo reaches the blastocyst stage. Embryos that reach this stage with good morphological characteristics have a high likelihood of implanting. However, not all embryos reach day 5 or 6 of development. There are several reasons why embryos may stop at any point in their development, but the most common reason is that embryonic development is halted due to some genetic anomaly within it. There are variations in the characteristics of embryo culture among different clinics and even among patients. However, they always agree that the conditions for embryo culture must be perfectly regulated, as any alteration can affect embryonic development and compromise the chances of success in fertility treatment. Regarding the types of embryo culture, depending on the duration of the culture, it is distinguished between short culture, generally up to day 3 of embryonic development, long culture up to the blastocyst stage. The same culture medium can be used throughout development, single medium, or several different ones can be used. Sequential culture. Whether one type of culture or another is performed depends on several factors, such as the number of eggs obtained, the quality of the embryos on day 3, or the need to perform a pre-implantation genetic test. Regarding the necessary conditions, the eggs and embryos must be in as stable an environment as possible. To achieve this, they are kept inside incubators for most of the time, as this prevents the culture conditions from being altered. Aspects such as temperature, concentration of carbon dioxide and oxygen, and humidity. In addition, it is also necessary for assisted reproduction laboratories to have very controlled conditions. Some of the factors that are regulated are air purity with very few suspended particles, light, temperature, and the gases present and in what percentage. Embryos develop in plates with culture media that provide the necessary nutrients for them to evolve correctly. As we have discussed, their needs vary at different stages, and therefore, depending on their composition, we distinguish. Sequential media contain the specific nutrients that embryos need at certain stages of their development. There is usually one medium for the first few days of embryonic development and another to use starting from the 3rd day. Single media contain all the nutrients that the embryo needs from day 1 of development until the blastocyst stage. Thus, the embryo will consume what it needs at each moment. And now we are going to answer the most frequently asked questions related to this topic. What is better, short culture or long culture? The reality is that the decision should be evaluated on a case-by-case basis. If we opt for short culture, the process is less complex and gives us the peace of mind that if we start with few embryos, there are more options to have one available at the time of transfer. On the other hand, extending the culture to blastocyst acts as a natural filter. It allows us to choose more precisely, since only the embryos with the greatest capacity to implant reach that phase. Ultimately, the ideal choice will be the one that best fits the history and unique characteristics of each patient, although the current trend is to transfer a single embryo at the blastocyst stage, long culture, due to the good results it offers. Are the eggs cultured the same way as the embryos? Broadly speaking, the environmental conditions are almost identical, although specific culture media are used to meet the particular requirements of the egg or the embryo. On the other hand, although both are extremely fragile, the egg is even more delicate, being a single cell with a very delicate internal structure. We would really love to hear about your experience, so we look forward to reading your comments.